Okay, we've talked about the tertiary structure of the fibrous proteins keratin and collagen, and now we're going to talk about the other category of tertiary structure, and that's the globular structure. A lot of proteins have globular structure, and that just means that it's not really easy to describe their structure. You can't just say it's a coil or a triple helix. It's just a combination of different secondary structures, and it looks like a big glob, right? Enzymes are typically globular proteins, and it's very difficult to figure out what their structure is. So before I show you an example of a globular protein, and we'll see another example in class, I want to tell you sort of how typically the structure of these globular proteins is discovered. Um, there are lots of techniques to try to figure out what the shape of proteins are. And NMR is a common one. You can look at the distance between atoms using NMR to get some basic structures of peptides or even proteins. There's another technique called X-ray dichroism, or sorry, circular dichroism that's used. But sort of the bread and butter technique, the gold standard, is an X-ray crystal structure. This is the same technique that Rosalind Franklin used on double-stranded DNA to determine the double helical structure, and the data that Watson and Crick used to assemble their models. And it can be used on proteins also. Now, the tricky part of this is in order to do an X-ray crystal structure on a protein, you have to have crystals of that protein. So you have to be able to purify the particular protein you're interested in and then put it under conditions so that crystals form. And sometimes that's relatively straightforward, and sometimes it's really, really difficult. Now, once you have crystals of proteins, you bombard them with X-rays. Those X-rays interact with the electronic environment of the protein and scatter in particular patterns. And these patterns can be read by a computer, and a structure of the protein can be inferred by the way the X-rays interacted and scattered with the crystal. Now, it has to be in the crystal because your protein has to be in this regular ordered pattern in order to give a regular ordered non-random X-ray diffraction pattern. Now, here's sort of the way you can build a protein by looking at X-ray crystal data. It's really you're looking at places of electron density, right, are going to diffract your X-rays more than open spaces. And so you can start to get a three-dimensional picture of a protein based on this X-ray diffraction. Let's look at an example, and we'll show another one in class. Here's a good example of a globular protein. This is an enzyme called acetylcholinesterase. It's important for really hydrolyzing or destroying acetylcholine after a nerve impulse to stop nerve impulses from continuing when they shouldn't. A lot of nerve agents, VX gas, for example, are inhibitors of this enzyme, and it's a good example of a globular protein. What's its shape? Well, it's a glob, right? And what's that glob composed of? Well, a lot of secondary structures. You can see some good alpha helices here and even some beta sheets, right? These arrows are indicating sheets, right? This looks like a parallel beta sheet, which is not that common. Here's maybe an anti-parallel. Well, wait, it's fine to get anti. What's this blue one up here? Here's an anti-parallel beta sheet with one chain of the sheet going this way, the other one going anti-parallel to that, and there's even a third one that's a third chain in that sheet that's interacting. And then there's some, secondary structures that aren't, really well formed. This blue ribbon that I'm showing here is probably pretty flexible. Okay, but the overall structure of this is a big glob. Now, oftentimes globular proteins, especially ones like acetylcholinesterase that are enzymes, they bind to some substrate, right? Acetylcholinesterase is a protein enzyme and it binds to acetylcholine. So oftentimes in globular proteins you can find a sort of a binding pocket, sort of a space or a cleft in the enzyme where the substrate binds. My guess it's probably right in here. It looks like there's a little hole in this three-dimensional structure of the protein. This is probably where acetylcholine esterase binds. Over here you can see sort of another depiction of the um, active site. This is the substrate binding pocket, and since this is an enzyme, it's likely that there's some activity in this site that breaks apart acetylcholine, hydrolyzing it.